All right, let's dive in. We're looking at a really interesting area today, making complex molecules.
Yeah.
Think, important stuff like life-saving drugs or intricate natural products.
Yeah, absolutely. And getting these right, getting them efficiently, often means you need to combine approaches.
Exactly. You need the best of both worlds, right?
Yeah.
Like traditional chemistry and the finesse of biology, using enzymes. We're talking hybrid synthesis.
Precisely. Nature's evolved these, well, incredible enzymatic tools, but sometimes you just need the sort of brute force or the specific transformations that only chemical reactions offer.
So blending them seems obvious, but it's tricky.
It is. It can unlock completely new, much better routes to really valuable chemicals.
But here's the snag, isn't it? The software tools that chemists and biologists use to actually plan these synthesis routes, they tend to live in separate worlds.
Mm-hmm. They're usually great at mapping out chemical steps or biological ones, but figuring out the best way to combine them, that's where they often fall short.
Which makes designing these potentially powerful hybrid pathways,
Yep.
well, incredibly difficult.
Yeah, it's like imagine you have a great map for highways and another for hiking trails, but nothing shows you the best place to park your car and start walking.
Good analogy. And that perfectly sets up our source material for this deep dive. It's a new research paper from JCSA introducing a tool specifically built to bridge this exact gap. It's called BioNavi.
Right, BioNavi, it's presented as a retrosynthesis planner, so it helps you work backward, from the molecule you want to figure out the steps and what you need to start with.
But the key difference is,
The key is it's designed from the ground up to consider both chemical and biological steps within the same search process. It doesn't just favor one.
okay, so our mission today, well, let's unpack how BioNavi actually does this. What's the magic under the hood? And why does the paper argue it's such a big step forward for planning these hybrid routes? Let's get into those details.
Well, what's really interesting, I think, is the way it combines approaches. It uses modern deep learning, but also integrates more traditional reaction templates.
Okay, deep learning and templates. Why both? What's the problem with just one?
Well, traditional tools often lean heavily on templates from reaction databases, right? But that means they can struggle if you need a really novel reaction not seen before. Deep learning, on the other hand, can suggest totally new pathways, but sometimes it feels a bit like a black box. Hard to know why it suggested something.
So they're trying to get the predictability of templates, but the novelty of deep learning. How's the combo work?
Exactly. They built a deep learning model, a transformer architecture, which is known to be pretty good at handling sequence-like data, like chemical structures. And they trained it on a massive data set covering both chemical and biological reactions.
Okay, but isn't there way less data available for biological reactions compared to chemical ones?
That's a key challenge they tackled. They used something called multitask learning. So the model learned to do two things at once: predict the precursors and classify the reaction type chemical or biological. And crucially, they used weighted training to give the biological reactions more importance, sort of balance things out despite the data difference.
Clever. So the AI suggests steps,
Hmm.
flags them as potentially chemical or bio. Then what? Where do the templates come in?
Right, so after the deep learning part proposes potential steps, BioNavi then checks. Does this suggested reaction actually match known reaction patterns in our template database?
Ah, like a reality check.
Pretty much. That template match helps estimate feasibility, makes the AI prediction more interpretable, and can even help suggest specific enzymes for the biological steps. The initial score from the AI gets adjusted based on how well it fits a known template.
That integrated approach makes a lot of sense. So the big question,
Yeah.
how did it actually perform? Did they test it against other tools?
They did, and it showed really strong results. On data sets of natural products, for instance, BioNavi managed a 97% success rate in generating complete pathways.
97%. That's high.
It is. And maybe even more compelling, it had a 75% hit rate in actually replicating reported biosynthetic pathways from the scientific literature. That was the best performance among the tools they compared it against.
Wow. Okay, replicating 75% of known bioroutes. That really speaks to its ability to find those relevant biological steps, doesn't it?
Absolutely. It strongly suggests the model has genuinely learned meaningful biological transformations, not just chemical ones. And crucially, for its main goal, it demonstrated a superior knack for designing hybrid pathways.
Ah, the core mission. How much better was it?
Well, on that natural product set, BioNavi predicted that nearly 65%, 64.7% to be exact, of the pathways it generated were hybrid. That was significantly higher than other tools, which tended to focus much more heavily on just chemical or just biological routes.
So that higher hybrid hit rate is really where it shines. It's tackling that specific planning problem we talk about. Did they show it working on, like, real-world drug examples?
Yes, they looked at drugs and natural products. For nirmatrelvir, that's an anti-COVID-19 drug, BioNavi came up with a pathway that closely matched the published synthesis. It even suggested a slightly different, potentially shorter order for some key steps, hinting at optimization possibilities.
Okay, so potential practical value right there.
Definitely. They also tested it on jamphthonic acid A, a natural product. BioNavi found hybrid routes starting from simple bio-based precursors, which lined up well with the recently published chemoenzymatic synthesis.
Aligning with published work is always a good sign.
And for simpler molecules like syringic acid, they showed it could be constrained to find purely biosynthetic routes, often finding simpler paths than older multi-step chemical methods. The paper really pushes this idea of streamlining discovery with enzymes.
And accessibility, is this just a research thing, or can people use it? They mentioned a web server.
They did. They've apparently improved the BioNavi web server to make it more user-friendly. It automates some parameters, integrates prediction tools for things like enzyme specifics or conditions. It even has a step-by-step mode.
What does that do?
It allows the user, the chemist or biologist, to guide the search with their own intuition and expertise. So it's not just fully automated. There's a layer of human control possible.
Right, combining human expertise with the AI's power.
Mm.
Okay, so let's pull this together. BioNavi sounds like a genuinely useful navigator for these complex hybrid synthesis paths.
It seems so. It successfully blends deep learning and templates. It definitely shows a strong ability to find those crucial hybrid routes, and the results on real-world molecules look promising.
But it's not perfect yet, right? The paper must mention limitations.
Oh, for sure. They're quite upfront about the challenges still ahead. They point out, the ongoing need for just more comprehensive reaction data overall.
More data, always more data.
Yep. Including what they call negative data, knowing what reactions don't work well or give low yields. That's super valuable for judging feasibility.
Right. Knowing the dead ends is just as important as finding the path.
Exactly. And they stress the need for more complete stereochemistry information in databases. That's critical, especially for biological reactions.
Huge for drug development, stereochemistry.
Absolutely. Plus, they acknowledge that things like predicting exact reaction conditions still need work. And critically, even with the best computer plan, you often still need significant enzyme engineering effort in the lab to make it actually work. The software doesn't solve the benchwork problems.
Of course. It's a planning tool, not a magic wand for the lab. Okay, so wrapping up, here's a thought for you, our listener, to chew on. What does a tool like BioNavi really signify for the future of making complex molecules? How does it fundamentally shift things, bridging that gap between designing a pathway on a screen and the, let's face it, often messy reality of making it happen in a flask or a bioreactor?